Lab report 6 deals with microbial growth. So what kinds of environments do we need to give bacteria in order for it to grow? And so there are several environmental factors that can affect the growth of bacteria. Things like temperature, oxygen, nutrients, pH, water. So we're going to explore several of these during this virtual lab. One of the first ones that is explored is oxygen. Now, this concept of oxygen requirement, we saw it in chapter 4 in lecture. So the reason why oxygen would be needed, and actually we just saw it last night on chapter 6 on metabolism. The reason why a microorganism will need oxygen is because oxygen is used as the final electron acceptor on an aerobic electron transport chain. So you can see down there how oxygen is taking up the electron that is being shuttled from one electron carrier to the next in an electron transport chain. And that is what causes the need for oxygen. Just trying to get my, let me do something real quick. I've got too much stuff open and it's just, okay, close some stuff here. All right, okay, so essentially what this is saying is that the organisms that will need oxygen are the ones that have an electron transport chain that needs oxygen, an aerobic electron transport chain. Those are the organisms that are going to need oxygen. We mentioned in chapter 4 and in chapter 6 that if you need oxygen, that requires the organism to have enzymes that will protect it from oxygen radicals. So if the organism has catalases and superoxide dismutases, or SODs, then the organism is well protected from oxygen requirements, from oxygen radicals. Okay, so the two enzymes are going to be catalase and superoxide dismutases. The ability of the organism to tolerate oxygen will depend on two factors, or there's two questions that you ask when it comes to oxygen requirements. Does the organism use oxygen? And does the organism have the enzymes that produce oxygen? So you can have an organism that uses oxygen, and those organisms are going to have the enzymes. If the organism does not use oxygen, the next question is, can it tolerate oxygen? Can it live in the presence of oxygen? And if the answer is no, then that means it does not have the enzymes that allows it to live in the presence of oxygen. So no, it is not using it, and it doesn't have the protective enzymes, therefore oxygen is going to kill it. The organism may have some of the enzymes needed, may have superoxide dismutases, but not catalases, in which case it is somewhat protected, but not completely. So those are the two questions to ask when it comes to oxygen requirements. So we, in chapter 4, we classified organisms based on their oxygen requirement as strict or obligate aerobes, which were the ones that needed oxygen and had the enzymes catalase and SODs. We have the strict or obligate aerobes, which do not need oxygen and do not have catalases or SODs. So it's not just a matter of not needing oxygen, oxygen kills them. Facultative anaerobes are going to use oxygen if available. If no oxygen is available, no big deal, they're not going to use it. They're going to turn to pathways that do not require oxygen. Of that, facultative anaerobes have catalases and SODs. So they're really equipped for any type of metabolism. We talked about aerotolerance in chapter therefore, these are organisms that do not need oxygen, but they have poor tolerance of oxygen because they only have SODs, they have no catalases. And then we also talked about the macrophiles that need oxygen but need a lower concentration of oxygen. They do have catalases and SODs, but they have them in smaller amounts, which means that they are not protected into the presence of atmospheric oxygen. So they need less oxygen, about 2 to 10% oxygen. Okay, so to find oxygen requirements, there are several procedures that can be followed. In the case of the virtual lab, there are two different techniques that are used. One technique uses a medium called thioglycolate. And let me go ahead and leave these and go to the, I'm going to go to the whiteboard, just explain thioglycolate a little bit better because I don't have it in the PowerPoints. So thioglycolate is a reducing medium. That's what it's described as. It is a medium that when it is boiled, it is going to get rid of the oxygen. And I'm not sure how it's spelled, so I think it's thioglycolate. So it will get rid of oxygen when boiled. In the exercise, you don't see it boiled, but that's essentially what you have to do. You have to boil it. In order to indicate the presence of oxygen, the medium has an indicator. Better yet, it's called an oxygen indicator that lets you know whether oxygen is present in the medium or not. It is called resasurin. And this is something that it is pink if there is oxygen present, and it is clear if there is no oxygen present. So the whole process is kind of straightforward in that you would boil the medium. The medium is a liquid. Obviously, in the virtual lab, you don't boil it, but let me just go ahead and draw two little tubes for a reason here. Okay. So if the tube looks pink throughout, then that tells you that there's oxygen present. You cannot use the tube because the idea of the tube is to have an oxygen gradient. You may have a little bit of pink on top because that is the area where oxygen is available. So the tube is a gradient. On the top, there's lots of oxygen. And as you move to the bottom, there's going to be less and less oxygen. So by the time you get to the bottom, there is no oxygen left. So the idea is to inoculate the medium with the organism. So you put the organism all over the medium, and then you incubate. Over time, if the organisms that are in the area where oxygen availability is optimum for the organism, that's where the bacteria will grow. So just to give examples here, if the organism is a strict aerobe, so we're talking about fluid thioglycolate medium. If the organism grows only on the top, and they can grow a little bit below the top also, not just strictly on the top, then you can say, okay, it only grows in the presence of oxygen, so the organism is a strict aerobe. If you have the organism growing only at the bottom, then you can say, oh, the organism is growing only away from oxygen, then it is a strict anaerobe. And you will see the presence of growth by cloudiness. So you will see that the tube is clear except below where there's cloudy, indicating the presence of organisms. If the organism is growing throughout the tube, top and bottom, then you can say, oh, this is a facultative anaerobe. So that's how you will read these tubes as to the requirement of the organism for oxygen. What we do in the face-to-face lab is we use, instead of fluid thioglycolate, we use what we call soft agar, which is a medium that has a very low concentration of agar, meaning that the medium becomes is slightly thick when is at room temperature. You boil it and the agar melts and it becomes completely liquid. So the idea will be the same as what we have here. I'm going to go ahead and go for the PowerPoints. The idea would be to, I'm just looking for the PowerPoints for oxygen. Okay, there we go. To take that soft agar deep, okay, obviously you always label your tubes. We boil it until it melts, then we cool it so that it doesn't kill the bacteria. Then it is inoculated using a 1 ml sterile pipette. We inoculate with 0.1 ml of the organism. We disperse throughout the tube so that the organism is all over the tube, and then we immediately ice it so that it becomes hard and the bacteria are trapped wherever you put it. So the bacteria are going to be all over the place and it will be trapped once the agar hardens wherever you put it. And so then you incubate, and wherever the cells fell in an area where the oxygen requirement was optimum for the bacteria, that's where you're going to see the growth. This is a nice view. This could also be a fluid thioglycolate tube, with the strict aerobe, strict anaerobe, facultative anaerobes. Macrophiles, I forgot to mention that one. Typically, macrophiles grow away from the surface, so there's no growth here on the surface, and there's growth away from it where there's less oxygen. Aerotolerants are the tricky ones because they grow throughout, so they may look at as a facultative, except the difference is with a facultative, you have tons of growth on top, and here you're not going to have any much growth on top. That's a difference. These are poor growers to begin with, so you're not going to see much growth. And the virtual lab does not work with aerotolerance anyway, but so that you'll understand what the difference is between a facultative and an aerotolerant. The other method of oxygen to test oxygen requirements is using an anaerobic jar. And an anaerobic jar is going to be a jar in which we're going to create an anaerobic environment. So the jar has you put your plates in here that you've inoculated with your organisms, and then you put a little packet that is purchased, and the packet is going to release hydrogen into the medium. The hydrogen is going to combine with oxygen, and it's going to create water. So over a period of incubation, you're going to see water droplets, because that's the condensation of the water that was created. Now, this is the problem. If you remove the oxygen from the inside of the chamber, now the pressure, the oxygen molecules now are in the form of a liquid, so there is less gas inside the chamber, and the air pressure has gone down, which means now the air chamber becomes a vacuum, and it would be very difficult to open the air chamber again. So to prevent that, the same package that releases the hydrogen is going to release CO2. And CO2 has two advantages. One is that it takes the place of the oxygen so that a vacuum is not created. And number two, some of the organisms, especially the anaerobic ones, like CO2. So they prefer to grow in high CO2 environments. So that kind of gives them a better environment for the anaerobic organisms. The other thing that is put in the jar to make sure that everything is working properly is an indicator, an oxygen indicator. The oxygen indicator that we use typically is methylene blue, and methylene blue is a dye. It is blue in the presence of oxygen. It is white with no oxygen. So this is a little strip of methylene blue, and when you put it in, is blue. And as you close the chamber and you let the packet begin to work, you can see how the little strip begins to change colors and becomes white. And that tells you the oxygen has been removed from the chamber, is now in the form of water. Okay. So in the anaerobic chamber, you would grow organisms in the anaerobic chamber, and you would compare the growth without oxygen with the growth of the same organism in a regular incubator with oxygen. So you heard the growth, and that should give you an idea of what the oxygen requirement of the organism is. So, for example, in the anaerobic jar versus incubator, let's say there is growth on both, and that tells you the organism can grow with or without oxygen. Chances are it's a facultative anaerobe. The only problem with this is that it doesn't allow you to distinguish between facultative anaerobes and aerotolerants. Both will grow in the same circumstances, but that's okay. It's not, a terrible thing. If the organism grows in the anaerobic jar, but it doesn't grow in the incubator, then you can say this is a strict anaerobe. If the organism does not grow in the anaerobic jar, but it grows in the incubator, then that's a strict aerobe. So that's basically how the whole process is going to work. Okay. All right. Any questions? Can we replace, the tables with a screenshot of your notebook for this lab? If the screenshot of the notebook has all the information requested, yes, absolutely. I have no problem with that. Yeah. And also, that will be fine. Now, make sure, however, that the screenshot of the notebook is not on top. Okay, I'll go into that one in just a second. Not in this lab, but it is on the report. Oh, I see. Okay, give me just a second. I'll address that. Yeah. Okay. Let's think. What was I going to say? Oh, yeah. Okay. Make sure that if you take a picture of the notebook, and the notebook or the results are not on top of the plates. So I want to see the plates by themselves. I want to see the notebook by itself also. Okay, so just make sure of that. Now, you reminded me of something, Joel, and I should have remembered, and I didn't. Pseudomonas aeruginosa was not in the lab, but it is a report. This is the deal. The organisms on the virtual lab change. So you what I need to do next time, too late for you guys now, but I need to leave the place where the names of the organisms are, I need to leave those blank, so you guys put the names of the organisms, because they may change. So not everybody's getting the same organisms. See what I'm saying? So I need to do that, and I forgot. So if you have an organism in the chart on the lab report that you didn't get, just replace it, okay? All right. So initially, I assume everybody got the same organisms. I have no idea. Honestly, I really don't know, because in order to know, I would have to do the virtual lab several times to see the organisms I get. Or I guess I could go back to lab reports from last semester. I mean, I haven't been doing these virtual labs long enough to have accumulated all that knowledge yet. So honestly, I don't know. Yeah, I don't know which are the organisms that are given. Labs don't all say that the names are only. Oh, got it. Okay, yeah, I see what you're saying. Okay. Yeah, I see what you're saying. So typically, the lab, what you can see is the name of the species. Oftentimes, the name of the genus is just a letter. So since you're taking pictures of the plate, you can use the same label that the plate has, like tea cape. I don't know what tea cape. I don't know what that one would be at all. Are you sure it's a tea? I have no idea what that one would be. That one doesn't ring a bell at all. So, it goes a picture. Okay. All right. It may not be a T. Yeah, I know. I see what you're saying. I see what you're saying. Okay. All right. I will. It was hard to see. Okay. All right. That doesn't help, does it? All right. Let's do these. And this was for the oxygen requirement, right? Imagine. Clip. Yeah. All right. So since you're giving me the picture of the, you're giving me the picture of the plate. I'm not gonna count off for that. Let's put it that way. Okay. I'm not gonna count off for the names of the organisms. Let's do that. And when I see the picture of the plate, then I'll try to figure out what the organisms are, and then hopefully, I will do better next time at giving you the names, and that way you don't have to be guessing like these. Okay. All right, so let me do this. Let me go ahead and go back to the, give me a second. Okay, got that. Okay. I'm just looking at the information provided. Okay, that's good. All right, temperature. I'm just looking at these. Okay, so we got the temperature. Temperature is a little bit simpler. I'm just looking at the lab report. Give me just a second. The most turbidity at what point the generation time is the shortest. So, okay. So when it comes to temperature, we're looking at the most organisms are going to grow at a range of temperatures. So they have a range of temperatures, not one set temperature, but it is a range of temperatures at which the organism is capable of growing. So part of, I'm just going back to, give me a sec. It's a PDF. All right. Okay, I'm just trying to go back to the beginning. All right, so within that range, there is a best growth temperature. One of the things about temperature is that it is, okay, there we go. The curve for temperature is a lopsided curve. And the reason why temperature is important is because it influences the activity of enzymes, and that's true of a lot of these requirements. So the range at which the organism grows is the growth temperature range, with the optimum temperature being the best temperature at which the organism grows. Now, the best temperature at which the organism grows is the one at which the organism grows the fastest, which means it has the smallest generation time because it's growing so fast. Medically important organisms will grow at 37 degrees centigrade, that's body temperature, and that will be their optimum temperature for growth. Anything above the optimum temperature or going too high past the optimum temperature is going to denature the enzymes of the organism and kill it. It will also destroy the lipid membranes. Temperatures below the optimum growth, however, just slow down the organism, but it doesn't stop it from growing. So a temperature curve will be lopsided in that the peak is not in the middle. The peak is closer to the maximum temperature, further from the lower temperature, meaning there is more room for lowering the temperature. There's more tolerance to low temperatures than it is to high temperatures past the optimum temperature. Okay, and that's because of enzymes being denatured. And based on the temperature requirement, microorganisms can be put in categories. And in the lab, what we do is we just put the organism in 6 different nutrient broths, and we let it grow at 6 different temperatures, and then we compare their growth. So an organism, so that means that yes, yes, absolutely. The optimum temperature will always be at the top of the growth range. It's not going to be the highest temperature though. The organism may be able to grow past the optimum temperature. Maybe if the optimum temperature is 37, the organism may still be able to grow at 42 degrees or 45 degrees centigrade. But not much more than that. 50 degrees will kill it, will not grow at 50. Whereas on the low temperature range, it may grow as low as 10 degrees centigrade. So, but yes, the OT is definitely closer to the top range, no question about that. So a mesophile, for example, the key to a mesophile is that it has an optimum temperature around 37 degrees centigrade. That's the key for a mesophile. Even if it may grow as low as 5, as long as it grows at 37, and that is its optimum temperature, it is a mesophile. A cyclotroph is one that has an optimum growth temperature at room temperature. So it will be lower than 37. It may still grow at 37, but it may not grow as well as 37 as it does at 30 degrees or 25 degrees. A thermophile will obviously grow at high temperatures. Okay. So in the exercise, you're given an organism and you figure out the temperature requirement of the organism by measuring the optical density. So in this case, you are not looking for growth, you are measuring the growth. So what you're doing is you're putting the organism in the spectrophotometer and seeing how much growth is there. Remember that the OD is a measure of absorbance. So essentially, the higher the OD, the higher the absorbance, the more growth you have. Okay, so if the organism has a higher OD at 27 than 37, so it has a little bit lower OD, okay, which means less growth, and even lower OD, so there is the tube is more clear, that means even less growth, then you know that 27 is the better temperature compared to 37 and 40. So that's the idea of using an optical density. Okay. pH is straightforward also. It's just a matter of determining at which pH the organism grows best by measuring OD. So again, you would expect most organisms, and that is the case, most organisms will definitely prefer a neutral pH. Most bacteria are neutrophiles. Just trying to get there. But pH of 7, that's what makes the organism a neutrophile. They may be able to grow a little bit more acidic, a little bit more basic, but in general, hovers around that 7 pH. Acidophiles will be the acid-loving ones, and alkalophiles will be the alkaline or base pH-loving ones. So pH, I think, is pretty straightforward. Osmotic pressure has to do with water availability. And remember that we said in chapter 3, I think it was, that bacteria are adapted to living in hypotonic environments. So they are adapted to living in environments that have a lot of water available. They are better equipped to handle water going into the cell than water leaving the cell. So hypotonic environments can be very damaging. Most cells prefer a wider environment or cannot withstand water environments any higher than about 0.85%, go as high as 0.9% salt. This is what we call a saline solution or a normal solution. This is the type of solution that our body is in. So when you taste tears or you taste sweat or blood is salty. That is this 0.85% salt. And that's what most bacteria like. That's the osmotic environment that they tolerate. So what that means is that a halotolerant is able to withstand higher salt concentrations. That doesn't mean that they like it. It just means they can tolerate it, which is very different. There is an important genus of bacteria, the Staphylococcus genus, which is very important medically, which is halotolerant. It can withstand high concentrations of salt. And so we use medium with high concentrations of salt to separate Staphylococcus aureus from other organisms, because all other organisms cannot withstand the 10% salt. To be a halotolerant is different from being a halophile. A halophile needs a high concentration of salt. So a halophile will have no growth in a 0.85% salt solution, whereas a halotolerant will definitely very happily grow at an 0.85 solution. Okay, so that's a difference. So something that grew at 1% would be halotolerant. Oh, maybe. I think 1%, depending on the exercise, if that's the, let me see what the exercise is. 1% is close enough to 0.85, 0.9, to where that may be what the exercise is taking as a normal solution. No. So 1% is taken as a 0.85%. Yeah, I wish they would have gone lower than 1% because a normal solution is lower than that, but that's okay. They gave you 1%, no problem. So this is close enough. So they're saying this is 1%. That's fine. Yeah, okay, there we go. Yeah, so definitely that's the 1% solution. So you would expect most bacteria to grow at those environments. 6.5%, it's getting a little bit too high. Definitely 10 is way too high. 25 is way too high. So only a true halotolerant will grow at a 25%. Give me a second. Somebody asked something. Yeah, halotolerant and intolerant. Okay, so the intolerant will be quote unquote the normal bacteria, the one that cannot tolerate high salt concentrations. So it would only grow at the normal solution, normal saline, which is the 1%. That's what they're giving you as normal saline. Technically speaking, it's 0.85 to 0.9. So yeah, so they're using intolerant, which is fine. Halophile, halotolerant. Yeah. So it should be straightforward, hopefully. No, you don't need to repeat the exercise. Yeah, your results should be fine. It's just that again, they're using that 1%, and most bacteria these halophiles are rare. These are organisms growing in extreme environments. Staphylococcus is not rare, however, and that is a halotolerant. So it's an important thing, piece of information to remember about Staphylococcus, that it can actually not only survive, but it actually it can be that can be used to separate Staphylococcus from all the other bacteria. And we do that in the lab all the time. Okay, so I wanted to go ahead and take a look at the lab report to make sure we didn't leave anything out. Okay. And I've already been looking at it. Okay. So we got the oxygen requirements, we got the anaerobic jar. This is the optical, this is the temperature, pH, osmotic pressure. Okay, so we'll just review and repeat that. What is the environmental environment most pathogen? Yeah, keep in mind what kind of environments pathogenic and opportunistic pathogens like, and essentially it will be any environment that human being like. That's basically it. And so when you look at what you can do to grow them, turn that around and think about what can you do to kill them. So, if there's a certain temperature that they like, then if you want to kill them, you want to increase the temperature past that optimum temperature in order to kill it. And same thing with pH or any other environment. Now, on the review section, can you explain how you want us to use? Yeah, the chart. Okay, my hope with the chart was that you could get the organisms that you work with in the virtual lab, and if you cannot read their names, put whatever it is you can read on their names, okay? So get as close at it as possible so that I can say, oh yeah, this is this organism. And summarize what you did on those organisms. Now, some organisms may just have been used for one test. So maybe an organism that was a halophile, that would be, let's say, I cannot write in my little thing, but the halophile one, no, it doesn't let me write. Let me try it again. Oh, it does let me write. Okay, so halophile organism A, you probably only used it for that exercise. So you would say it's a halophile. Okay, so that's the only information you have for that organism. Other organisms, maybe, I'm just going to guess here, I may be completely wrong. Let's say E. coli may have been used for oxygen requirement. So you may know, its oxygen requirement. You may use for the temperature. So you say, okay, mesophile. And then it says catalase. Okay, well, I didn't test for that. What are they talking about? Well, if you know the oxygen requirement, you should be able to tell me whether the organism has catalase or SODs. So if it is a facultative, then it will be positive for both, for example. Okay. So the answer for catalase or SOD comes from the oxygen requirements. So just take the organisms that were used on each of the exercises and give the information that you gather from the exercise on that organism. If you need more room, just create more room. And again, most of the organisms, you may only have one piece of information for them. Maybe Staph aureus was only used for the temperature, maybe. The way that we are able to go back in the lab to get the picture. Ooh, that's a good question, and I do not know the answer. To go back to get the pictures. Let me see. To back in the lab to get the pictures for our lab report. I have closed out before I was able to get the picture on the screenshot. Oh, okay, your browser cache. Oh, really? That will do it? So if you were to go on your browser, you should be able to get to that again. Okay, Joel is saying that hopefully that does it. Other than going back and repeating the exercise, which, and the other option you would have is just call McGraw-Hill and see if they can do it, or they can tell you how to do it. Yeah. Okay. So if the browser will do it, then that will be great. Yeah. Does that help, Mercedes? Hopefully it does. Okay. Any other questions about this lab? Hopefully these labs are getting easier. That's my hope. Oh, you finished the lab? Oh, Okay, so like I said, hopefully these labs are getting a little bit easier. We're going to meet on Sunday to go over the okay, normal for a growth range to be pH 5 to 9. I would say less than 9. 9 is pretty high. 5 is okay. 9 is pretty high. Fungi are considered neutrophiles, and yet they grow at 5.5. That's what they really, really like, and yet they're classified as neutrophiles. So we say they like a slightly acidic environment. So yeah, 9 seems a bit too high to me. But yeah. So I think for a neutrophil, I think the range is about 5 to 8 or something like that. Yeah, one of my bacteria grew at that range, but did it grow better at 7 or around 7? And it does seem pretty high. But if that's what you got, that's what you got. It's fine. Oh, huh, interesting. So the absorbance did not change. Oh, interesting. So the absorbance was the same across from 5 to 9. Interesting. Okay, according to the graph. Okay, all right. And the graph was done using the absorbance. Okay. So you have the absorbance values. Okay. All right. All right. If it is that's what you got, that's what you get. It's fine. Um, Unfortunately, these are virtual labs. And if it was a real lab, I would say, okay, did you mix the tube properly? Did you, I assumed you zero. I think the machine makes you zero. Hopefully you zero it properly. Yeah. Okay. Good deal. All right. That's fine. If that's what you get, that's what you get. Yeah, they do all the work on this. Kind of annoying, actually, if you ask me. But okay. Yeah, the little thing does everything for you. That okay. I know. Okay, that's fine. If that's what you get, that's what you get. More than anything, I want the concept understood. That's really what I'm after more than anything. So we'll go over the study guide. I'm going to be posting the study guide by Sunday morning, and then we'll go over it Sunday evening. And that way, starting Monday, if anybody's ready to take the practical, I'll open the practical, and you have until I think it's Saturday to take the practical. And there is no lab report. Yes, I can answer questions about the lecture test. Okay, sure. Let's take about 5 minutes for that. I do not have a lot of time, but yeah, we can go ahead and do it. Give me a second. Okay. You either open microphone. Oh, there we go. Give me a sec. Well, we talked about fluoride. Or is the same thing? It goes together. Fluoride is a non-competitive inhibitor. So 2 questions about it is if it is a non-competitive inhibitor that indiscriminately inhibits enzymes, what would be its selective toxicity? Would you say it has a good selective toxicity, or would you say it has a bad selective toxicity? And yes, correct. Right. So it is going to be poisonous to humans and to bacteria. The difference is bacteria are tiny, we're big, so we can withstand more chloride or fluoride, sorry, than they can. Having said that, we don't let little children brush teeth with fluoride toothpaste because that is because we know that they're little and they're little, they may not be able to withstand as much fluoride poison as an adult would. It is a poison, period, and we've been putting it in water forever. And there's a huge lawsuit that was just won, I think it was at the beginning of this year in San Francisco against the EPA for fluorinating water for all these years. So that may be changing hopefully sometime soon. So yeah, it is definitely, it has no selective toxicity. It has very poor selective toxicity. So, no, toothpaste is one thing. We're just brushing your teeth and then spitting it out. We're not eating it. Drinking fluorinated water is a different thing. It's equivalent of drinking a sunscreen instead of putting it in your body. So anyway, yeah.
Real quickly, last question. Can we go over the exergonic, endergonic, catabolic, anabolic part of ATP synthesis? Like how I thought that phosphate breaking, like when you add a phosphate, it's endergonic because you're adding the energy to.
Yes, because you do that.
And then when you break
Yes, absolutely.
that's when it's exergonic.
However, you need energy to do that, and the energy is coming from a catabolic reaction.
Oh.
I'm trying to get out of here so I can get a whiteboard. So that's why the two are so linked, is because let's see, at the end of okay, so yeah, that's okay. Give me just a second. And I'm making this up. Is it is it here that we do this? Oh, no, I don't know. Okay, I won't give this example. Yeah, it's okay. All right, so let's say in glycolysis, we have a couple of steps. No, it's okay. I do understand, and actually, you make a good point. I need to go back to the PowerPoint and fix that where it links AT synthesis with catabolic reactions, because it is linking it in a way that is not clear. So during glycolysis, which is a catabolic pathway, there is a point in which a phosphorus is removed from a molecule and energy is removed as that bond is broken. That is used to take ADP and make ATP. So when you look at these, this right here is a catabolic reaction and it's exergonic. However, if you look at the making of the ATP, that is anabolic and is endergonic. So you can look at it as two reactions happening at the same time, one catabolic, one anabolic. Does that make a little bit more sense?
So catabolic breaks it, and when it breaks it, it releases the
energy.
It releases the energy, and the energy is stored in the phosphate bond. And then when the released energy, when that released phosphate bond is added to adenosine triphosphate, that is anabolic.
Correct. Yes.
And that's endergonic, making of ATP.
Exactly. And you're creating a bond, ATP and the phosphate.
Because that's where the energy is stored, is in the bonds.
Exactly. Exactly. Okay. Yes.
Thank you.
Yeah.
Stop right there before I confuse myself again.
Yeah, there you go. No. But that's a really good question, and you make a very good point that I had never. To me, it was such a given that I had never thought about it with that much detail. So I need to correct the PowerPoint so that I don't link the two and make it confusing again.
The way I learned it from Professor Siegel was anabolic bodybuilders get bigger, and marathon runners get smaller because they have catabolic. Their bodies undergo catabolism.
Yes. Mm-hmm.
And so that's been my thing in the head. Anabolic makes them bigger, and catabolic makes them scrawny.
Well, you made it bigger. You went from 2 ADPs to 3 ADPs.
Yeah. So that's anabolic.
Absolutely. Yep. Okay. Yep, you got it. Very good.
Stop right there.
Yeah, there you go.